to the lecture series in animal cell culture. So, we are into the week 6 and we are starting our 4th lecture. So, if you remember in the week 5, the last lecture, this is where we stopped, and I told you do not lose sight of it because we will be coming back soon after a bit of a digression of understanding the myelination. So, you have adult hippocampal dissociated cells, you have pyramidal cells, you have interneurons, you have astroglia, you have oligodendrocytes. So, now how one can separate these cells out when you get an adult tissue. This is where we started, right. So, this is what you are going to get, a piece of tissue which is the hippocampal neuron. Okay. So, the way you are achieving it is that first of all you have to isolate the individual cells and you have to re-establish the contact. So, this is the population what you are getting: pyramidal cells, interneurons, astroglia, oligodendrocyte. First of all you dissociate these cells and we have talked about the dissociation protocol. Next, once you have dissociated these cells, how you can separate out the cells. So, one of the ways, now I will come to that. Now, we are into week 6, okay. Lecture 4, W6 L4. This is where we are. Now, when you talk about getting the adult hippocampal neuron and how we are going to separate, so again just let me enlist pyramidal neurons, interneurons, oligodendrocytes, you will have microglia, you will have astrocytes. So, one of the simplest way of cell separation is called density gradient centrifugation. Density gradient centrifugation. What really is density gradient centrifugation? So, each one of these cells, first of all, when you isolate the cells, try to see when you dissociate them, what are the size difference in the population. So, say for example, when you talk about the pyramidal neurons, these will be after dissociation when it has lost all its, if it is an adult, right, we are talking about the adult. So, it has lost all its processes, this will be something like this. If you see an interneuron, the contrary, you will see slightly smaller, whereas the oligos, if you look at them, they will be far more smaller. Microglia, you will not be able to see it really, this will be so small, and then you have the astrocytes, which are kind of similar to oligodendrocytes. And interestingly, in the process of development, these have formed at different time. So, in other words, in the process of development, these have formed at different time points. Okay. So, their requirements are also different. Now, one of the easiest way to go for it is, so each one of these cells will have different densities. based on their density, which is essentially mass is to volume ratio, you can separate these cells. What one has to do is one has to create a density gradient column. So, what is the density gradient column? So, you have some neutral liquid, something, it could be sucrose, it could be one of the new ones which are being used for last 10 years is OptiPrep. So, there are several such molecules which will not, sucrose has certain issues with osmolarity though, as compared to OptiPrep, which is much more easy to deal with. You form a density gradient. Density gradient means, so you layer the, so you can make these solutions of different densities. Say for example, you take OptiPrep, density 1, density 2, density 3, by mixing it with some suitable buffer, D5, D6. So, let us take this simple example. Okay, I make 4 densities, 6 different densities of, I take OptiPrep. I am not taking sucrose for a reason. There are issues and I do not want to get into that. I take X amount of OptiPrep and I use a buffer where the neuronal cells will survive, and Y amount of buffer. So, I mix them at a particular ratio and I achieve a particular density. Say I say density 1. Now, I mix them in another ratio, I achieve a density 2, and assuming density 1 is more than density 2. So, it means density 2 what I am getting is lesser dense. Okay. Then I make something density 3. So, now density 1, density 2, density 3. Then I get another one density 4, and I am just mixing them, okay, a small amount, maybe 1 ml thick, just 1 ml, 1 ml. So, then I get density 1, density 2, density 3 and density 4. So, let us just stick to 4. Okay. So, now I take a pipette, and on a pipette I layer the highest density 1 ml layering, and this layering has to be done very, very gently. So, density 1 on top of, and while you do this layering, this took me a long time to learn it. It is absolute by trial and error. So, whenever you are pipetting it, generally the way we do it, you hold the pipette and you just dump the fluid out there. But actually this density gradient should not be done like that. The way one should do this, make this density gradient is something like this. You tilt the whole thing like this, almost at a very, very acute angle. The more the angle is kind of close into 0, like if you can really hold it, and this comes by practice. There is no other, there is no science in it, or almost like this. That will be the best. The more the acute angle is, you cannot make it horizontal because it is going to flow out. And using a very narrow bore pipette here, you very gently allow the stuff to form that layer, something like this. So, this is where your pipette tip is. You can further go down actually if you have that margin, and slowly you layer it. So, what you achieve is that this is the layer 1, and you have to always keep it tilt, slightly slowly allow it to settle down, then put the next layer. This is your, if this is density 1, this is density 2. Then you put on top of that density 3, and it has to be done very, very gently. Trust me, this is not something very easy. Then on top of that, you achieve what you, so one second, density 3. Then you have density 4. So, you are layering different densities where the highest density is lying here, second highest here, third highest here, fourth highest. So, on the top you have the lightest of all. Now, on top of that you have these dissociated cells, right. You have dissociated these different kind of cells in a particular buffer. So, and I told you, you make these layers of, so for example, layer 1, layer 2, layer 3, layer 4. Okay. So, you make these layers. On top of this layer, you take, say, if you make this layer of say 4 ml or 6 ml, whatever, that I will leave it up to you, because it depends on cell types you are using. If you are using the pancreatic cells, you have to separate the, islets of Langerhans and all those things. You needed a different kind of layering, but the logic is the same. Or you are using the liver cells, or you want to separate out the cells of the lungs or kidney. You need different concentrations, but the logic of density gradient centrifugation remains the same. And the reason for me to pick up the neuronal case, because those are the last one in the history of this kind of density gradient centrifugation where they are being used. The earlier density gradient centrifugation has been used for other cell types. So, on this density gradient column, so what you make, what I asked you, that you make a density gradient column. And what is a density gradient column? This is the density gradient column what we created now. On top of the density gradient column, you have this dissociated cell, you make a very, 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 very thin layer on top of it. So, these are the cells which you want to separate out, and it has a mix of bigger cells, smaller cells, even smaller cell, smaller cell. Now, one more thing, like I will not ask you really to go and figure out what is the densities, but people over the years, almost last 50-60 years since this technique is there in the market, people have a fairly good idea about respective densities of the cells where they will fit in. Now, you take this, you seal the lid of it, and you spin it in a centrifuge. Now, while you spin it in a centrifuge, what will happen is, and please be cautious of having this topmost layer of the cellular, this thing has to be exceptionally narrow band. There are people who recommend thicker band, but over the years I have realized that that really does not work, and you spin it. Once you spin it, what will happen? Depending on the density of the cell, they will settle down at different density zones. So, in other words, if I have a density column like this of say D1, D2, D3, D4, D5, D6, D7, D1, D2. So, I am not following that pattern, I am just kind of for your understanding, I am D5, D6, D7. Now, the cells of different densities are going to settle down where they find their matching density. In other words, if you really master this technique of density gradient centrifugation, depending on the size or the volume of the cell, size includes here of course you can separate out the cells. This is one of the simplest, cheapest and easiest, but that needs a whole lot of optimization protocol. You really have to go through the rigor of optimization, but the basic fundamental of density gradient centrifugation is this. Now, if you have a hang on this technique, if you really understood the technique, the next part is, now you understand the basic physics behind it. Okay. Now, there is a practical side of the story which I haven't talked about. Okay, now we have separated the cells. Great. Now, once you separate the cells, now you have to pull them out. So, what you will be seeing is at every level, so you may see a band forming out here. Now, how to pull out that band? This is not easy. So, for example, I see 3 bands, 3 different zones. Now, you need something like a very similar to Hamilton syringe, which is used in biochemistry labs. It's not really Hamilton, but something of that sort, where you have to, bring it close in, and you have to pull out that band in a separate test tube. Similarly, then you have to pull this out at a separate test tube. You have to pull this out at a separate test tube. Once you see these cells out, so you have these cells of band, say this is the band 1, this is band 2, this is band 3. Now, when you are doing it for the first time, you have no idea which cells are which. Now, what you have to do, you have to characterize this cell by 2 methods, immunocytochemically is one method, and you have to grow them to see their morphology, how they grow over a period of time. Based on that, you will be able to figure out which bands represent what cell type. So, these are some of the technologies which has been achieved over the years, like the very first technology. I'll come after the hippocampal neuron, I'll come about the spinal cord neuron, where you have to separate out the oligodendrocytes and several other cell types there. I'll come to that. Okay. So, what I wanted to highlight here, so these are the basic technologies and the follow up technologies what one has to master before you achieve the goal of developing the defined systems. Earlier, for embryonic neurons, so this particular technique, so there was another such thing which is called nicodens. You won't find that anymore because they have discontinued this product. It is very similar to OptiPrep, nicodens, OptiPrep, sucrose. You can use sucrose also, but the sucrose, the problem is what I was trying to tell you, because most of these cells had a certain degree of osmolarity, and sucrose kind of, because of this molecule, it kinds of ruptures the cell at times because it creates an osmotic disbalance. But if you are really clever, very careful, you can achieve it, but I will not recommend unless you have mastered the technique beyond doubt. So, I would rather request you try something like OptiPrep or nicodens if you get it, but most likely you are not going to get nicodens, because nicodens has been discontinued, if I remember, almost 17 years back by Amersham and all those other groups who used to produce nicodens. So, but these are the different substrate, you can call them, to achieve a density gradient centrifugation. There is another way you can do this, which I will be coming in my next class, that how you can separate cells. So, as of now, in terms of the OptiPrep separation, what all has been achieved. So, these are some of the literatures I will be giving you. You can separate out the adult hippocampal neurons. So, now you remember where we left last week, and I told you I will come back to that, adult hippocampal neurons, and this is where we will be referring to Brewer's work, Gregory Brewer's work. This has been achieved for adult spinal cord neurons. There I will be referring to James J. Hickman's work. His group has pioneered these techniques Earlier to that, there was a very nice paper, very early, back in 1982 or 83, of, we talk about the adult, there are people who separate out embryonic motor neurons. I haven't talked about that. I'll talk about very soon in the next class on it. Embryonic motor neuron separation. I will hand over this, if I could get a copy of this paper, because this is a very old paper where all these density gradient centrifugation has been used. So, this is I'd say 80s, this is in the early 2000, and this is around 2003. So, this is how this area of cell separation in terms of the neuronal cell has progressed. Okay. So, I will close in here before I move on to the next class, where we will talk about the other couple of techniques of cell separation.